Hello, and welcome to this Anatomy Physiology Bio 202 video lecture on metabolism and nutrition. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. Metabolism refers to all the chemical reactions taking place in the body. Reactions that break down complex molecules into smaller ones are referred to as catabolic or decomposition reactions. Reactions that combine simple molecules to make complex molecules are referred to as anabolic or synthesis reactions. Metabolism results from the balance of anabolic and catabolic reactions. ATP, or adenosine triphosphate, is the energy molecule that couples the two types of reactions. This picture illustrates the connections between anabolism, catabolism, and ATP. Be sure to note what goes into each reaction and what comes out. Energy transfer. There are a number of reactions that we'll look at as we go through this material. One of the more important reactions to make sure you understand are the oxidation-reduction reactions. Oxidation and reduction reactions involve the transfer of electrons from one atom or molecule to another. Illustrated on this slide is oxidation, which involves the removal of electrons from an atom or molecule, and as an example, we see the conversion of lactic acid to pyruvic acid. Here, reduction is illustrated. Reduction involves the addition of electrons to a molecule. An example is the conversion of pyruvic acid to lactic acid, illustrated on this slide. When a substance is oxidized, the liberated hydrogen atoms are transferred by two coenzymes to another compound. These are nicotine adenine dinucleotide, NAD, and flavin adenine dinucleotide, or FAD, illustrated here. Oxidation and reduction are always coupled. That is why the two reactions together are called oxidation-reduction or redox reactions. The oxidation of lactic acid to pyruvic acid and the associated reaction of NAD may be written as seen on this slide. Some of the energy released during oxidation reactions is captured when ATP is formed. A phosphate group is added to ADP in what is referred to as a phosphorylation reaction, along with energy to form ATP. A high energy bond is indicated by a squiggly line as seen on this slide between the phosphate groups. Carbohydrate metabolism. Carbohydrate metabolism is, in reality, mostly glucose metabolism. The body's use of glucose depends on the needs of cells. These needs include ATP production, amino acid synthesis, glycogen synthesis, and triglyceride synthesis. Glucose must pass through the plasma membrane to be used by the cell. Facilitated diffusion makes this happen. In most body cells, GLUT molecules or transporters perform this function. Insulin increases the insertion of GLUT4 transporters into the plasma membrane, increasing the rate of facilitated diffusion. The oxidation of glucose to produce ATP is referred to as cellular respiration or aerobic respiration. There are four sets of reactions that are involved in this process: glycolysis, the formation of acetyl-CoA, the Krebs cycle, and the electron transport chain. This graphic illustrates each of those aforementioned steps and the products that they produce. Glycolysis is the process whereby a 6-carbon glucose molecule is split into two 3-carbon molecules of pyruvic acid. Glycolysis involves 10 reactions. Here we see where glycolysis fits into the big picture of the entirety of cellular respiration and the first few steps. This graphic illustrates the process of glycolysis using molecular diagrams. Here you can see glucose going through two phases of glycolytic reactions, first starting off as glucose, finishing as glyceraldehyde 3-phosphate in step 1, And then in step 2, the glyceraldehyde 3-phosphates are converted to pyruvic acid. Be sure to note what goes into and out at each step of this process. What happens to pyruvic acid after glycolysis depends on the availability of oxygen. If oxygen is scarce, we are in what is described as anaerobic conditions. If oxygen is scarce, pyruvic acid is reduced by the addition of 2 hydrogen atoms to form lactic acid. If oxygen is plentiful, we are in what is referred to as aerobic conditions. If oxygen is plentiful, most cells will convert pyruvic acid to acetyl-CoA. This diagram illustrates where the acetyl-CoA conversion step will occur. Glycolysis, converting glucose to pyruvic acid, occurs outside of the mitochondria in the cytoplasm in the absence of oxygen and in the absence of any special cell machinery. At its completion, if oxygen is present, then the pyruvic acid will be converted to acetyl-CoA. This graphic illustrates the next step in carbohydrate metabolism as acetyl-CoA enters into the Krebs cycle, also known as the citric acid cycle, provided oxygen is present. Here we see the Krebs cycle, also known as the citric acid cycle. This cycle occurs in the matrix of the mitochondria and consists of 8 separate reactions. Here you see a generalized diagram of the Krebs cycle. At its inception, you'll notice pyruvic acid being converted to acetyl-CoA. Acetyl-CoA enters the Krebs cycle and undergoes a series of reactions that oxidizes fuel molecules to reduce electron carriers. Those electron carriers will be used in the electron transport chain. This is a slightly more detailed diagram of the same process. The electron transport chain is a series of electron carriers in the mitochondria. Each carrier in the chain is reduced as it picks up electrons and oxidized as it gives up electrons. Exergonic reactions release energy used to form ATP. This mechanism links chemical reactions with the pumping of hydrogen ions and is known as chemoosmosis. This picture illustrates the location of the electron transport chain and, in general, summarizes the moving of hydrogen ions to create a chemoosmotic gradient that will provide the power to produce ATP. Electron carriers include flavin mononucleotide, cytochromes, iron sulfur centers, copper atoms, and coenzyme Q. Inside of the inner mitochondrial membrane, the carriers are clustered into 3 complexes, each acting as a proton pump that expels hydrogen ions. Here we see a more detailed depiction of the electron transport chain. Note the electron carrier, NAD, delivering electrons that were harvested from the Krebs cycle's fuel molecules or possibly the process of glycolysis. As those electrons are delivered, they're picked up by the embedded electron carriers. Those electron carriers will then transport hydrogen ions, again to create the chemoosmotic gradient that will be used to make ATP. Cellular respiration will generate either 30 or 32 ATP molecules for each molecule of glucose catabolized. The reaction is illustrated for you on this slide. This diagram gives you a complete overview of the process of glycolysis through the electron transport chain. And this table will provide you with a summary of ATP produced in cellular respiration. Note the source and ATP yield per glucose molecule. Glucose, not needed immediately, is stored as glycogen. This process is the one that creates, or the one that we describe as glycogenesis. When ATP is needed for body activities, glycogen is broken down by a process called glycogenolysis. This graphic illustrates that process of converting glucose into glycogen. Glucose may be formed from proteins as well as the glycerol portion of triglycerides, lactic acid, and certain amino acids. The process is known as glucogenesis. Cortisol, glucagon, and thyroid hormones stimulate gluconeogenesis. This diagram illustrates the process of gluconeogenesis, which is stimulated by cortisol and glucagon. Lipid metabolism. Because most lipids are nonpolar or hydrophobic, they do not dissolve in water. Because the blood plasma is over 90% water, lipids must be transported, combined with proteins, produced by the liver and the intestines. These are called lipoproteins. This is an illustration of a lipid protein. There are 4 classes of lipoproteins: chylomicrons, which transport dietary lipids to adipose tissue, VLDLs, which transport triglycerides from hepatocytes to adipocytes, LDLs, which carry about 75% of total cholesterol in the blood and deliver it to cells, and HDLs, which remove excess cholesterol from the body cells and the blood and transport it to the liver for elimination. Cholesterol comes from some foods, eggs, dairy, organ meats, but most is synthesized by hepatocytes. Increases in total cholesterol levels are associated with a greater risk of coronary artery disease. Exercise, diet, and certain drugs are used to reduce high cholesterol levels. Lipids may be oxidized to produce ATP. If the body does not need lipids at any given time, they get stored in adipose tissue. Some are used as structural molecules or to synthesize other essential substances. This table summarizes the types of lipids in the body. You'll note the lipid type and function listed. Adipose tissue is used to remove triglycerides from chylomicrons and VLDLs. These triglycerides constitute 98% of all body energy reserves. Lipolysis, or lipid catabolism, is a process of splitting triglycerides into fatty acids and glycerol. Lipid anabolism, or lipogenesis, is the process of synthesizing lipids from glucose or amino acids. It occurs when individuals consume more calories than are needed. This diagram illustrates the processes of lipolysis and lipogenesis. Protein metabolism. Digested proteins are broken down into amino acids, which are not stored but are either oxidized to produce ATP or used to synthesize new proteins. Many proteins function as enzymes. Some are involved in transportation, serving as antibodies, clotting blood, being hormones, or being part of muscle fibers. This table will summarize the functions of proteins. You'll note the type of protein that's listed and their function. Protein catabolism is the breaking down of protein. Breaking down protein yields amino acids, which are converted to other amino acids, fatty acids, ketone bodies, or glucose. Cells oxidize amino acids to generate ATP via the Krebs cycle. Protein anabolism, or protein synthesis, creates new proteins by bonding together amino acids on ribosomes. This is a depiction of protein metabolism. Note that each individual amino acid has the potential to enter the metabolic pathways we've learned about previously in different locations. Key molecules at the metabolic crossroads. Of the thousands of different chemicals in cells, glucose 6-phosphate, pyruvic acid, and acetyl-CoA are extremely important in metabolism. Glucose 6-phosphate is involved in the synthesis of glycogen, the release of glucose into the bloodstream, the synthesis of nucleic acids, and glycolysis. Pyruvic acid is involved in the production of lactic acid, the production of alanine, and gluconeogenesis. And acetyl-CoA is involved in helping 2-carbon acetyl groups enter the Krebs cycle and the synthesis of lipids. This is an illustration of the key molecules at the metabolic crossroads. And this table will summarize metabolism for you. Metabolic adaptations. Regulation of metabolism depends on chemicals in the cells and signals from the nervous and endocrine systems. Some aspects of metabolism depend on time elapsed since the last meal. During the absorptive state, glucose is readily available. This is a depiction of some metabolic adaptations. And this table will provide you with a summary of hormonal regulation of metabolism in the absorptive state. Note the process, the location, and the main stimulating hormones. During the post-absorptive state, energy needs are met by fuels already in the body. This is an illustration of the metabolic adaptations for post-absorptive state. And this table will provide you with a summary of the hormonal regulation of metabolism in the post-absorptive state. Again, note the process, the location, and the main stimulating hormones. During fasting and starvation, the body must make metabolic changes to survive. Fasting is going without food for several hours or a few days. Starvation is going without food or inadequate food intake for weeks or months. The most dramatic metabolic change occurring with fasting and starvation is an increase in production of ketone bodies as catabolism of fatty acids increases. They may be used for energy by all cells. The rates of metabolic reactions control the amount of heat produced by the body. The rate of heat loss must equal the rate of heat production to maintain homeostasis of body temperature. The metabolic rate is the overall rate at which metabolic reactions use energy. Your metabolic rate is measured with the body in a quiet, resting, and fasting state. This is your BMR or basal metabolic rate. Factors that affect your metabolic rate might include, but are not limited to, exercise, hormones, your nervous system, body temperature, ingestion of food, age, gender, climate, sleeping, and malnutrition. Heat can be transferred from the body to the environment by conduction, convection, radiation, and evaporation. The preoptic area of the hypothalamus is the body's thermostat. Thermoreceptors send information to the preoptic area, which sends signals to the heat-losing center and heat-promoting center of the hypothalamus, depending on the body's needs. Negative feedback mechanisms conserve heat and increase heat production as needed. Heat production is balanced through a negative feedback loop, as illustrated here. Finally, energy intake is directly dependent upon the amount of food consumed, and your total energy expenditure is based on your BMR, your physical activity, and your food-induced thermogenesis. And this concludes our coverage of metabolism and nutrition.